AssalamuAlaikum friends, welcome back to the channel. Today we are going to talk about Bacterial Lab Diagnosis. This is really crucial in understanding which infection is caused by which bacteria. Either our normal flora is involved or there's some foreign bacteria that got into our body and is responsible for causing infection. But before starting the lecture, I like to tell you guys that these videos are meant for educational purposes. Things and treatments may change with time. If I get wrong or miss anything, your input is always welcomed in the comments section. Grab a cup of tea and let's get started. Bacterial Lab Diagnosis. Bacterial lab diagnosis is an important tool in the diagnosis and treatment of bacterial infections and is often used in conjugation with other diagnostic methods such as clinical examination and imaging tests. The process of using laboratory methods to identify bacterial infections. Bacterial lab diagnosis involves analysis of the samples such as blood, urine, sputum, and stool. And there might be some other samples, but for your ease, I've put just these samples. It identifies bacteria that are responsible for causing the infection and determines their susceptibility to different antibiotics. It means that it guides in the treatment. Some bacterial strains are resistant to certain antibiotics, so by doing a bacterial lab diagnosis, we will know that which antibiotic is not useful against this bacteria, so we'll switch to another bacteria to treat that infection. Lecture outline. I've introduced you guys to the bacterial lab diagnosis. Now we are going to have a deep look at the methods, and at the end, as usual, we'll review the lecture. Laboratory diagnosis. The laboratory diagnosis of infectious diseases include microscopic, culture-based, immunologic or serologic, and molecular, which include nucleic acid and protein-based tests. Bacterial lab diagnosis involve following methods. But prior to doing any of the methods, we will collect the sample. This step is called collection of specimen. It is followed by microscopy. If microscopy is enough to diagnose an infection or tell us the cause of that infection, then we'll not go further. And if it fails to do so, then we'll go for culture. Same goes for culture. If culture fulfills our requirement, we'll stay with culture. If culture doesn't fulfill our requirement, we'll go for serology. If serology fails to fulfill our requirement, then we'll go for molecular diagnostics. Serology. Collection of specimen. The first step in the lab diagnosis of the bacteria. We'll collect a sample from the body side. There are two types of body sites. One is sterile, one is non-sterile. Sterile is something like that is not contaminated with any of the foreign organism. If we are taking a sample from urinary bladder, so urinary bladder is sterile from inside. There's no bacteria that is going to make this area contaminated or our sample contaminated. Non-sterile. For example, we are collecting a sample from a wound on a skin cut. So there might be certain foreign particles like soil, dust, and pathogens from the environment, atmosphere, and also from the skin around the wound. So this is the non-sterile body side, and the urinary bladder in that example I mentioned was the sterile body side. Then we will collect an adequate quantity of that sample for the contamination from the normal flora. If it is contaminated with the normal flora, it will be difficult for us to diagnose whether the normal flora is responsible for causing that infection or the infection is caused by the organism present in the sample. Integrity of that sample is not compromised during transport. Specimen is handled carefully, and ancillary information, which means the supplementary information, is also provided. There are different specimens collection site. I've mentioned following: blood, throat, sputum, CSF, stool, urine, genital tract, wound, and abscess. Microscopy. Prior to knowing what is microscopy, we should know what is a microscope. Microscope is a device that magnifies, means zooms in an image of an object so that it can be examined in more detail. And by using microscopes to view and investigate objects that are invisible to human eye or naked eye, this process is called microscopy. Types of microscopes. There are different types of microscopes, each with their own advantages and limitations. Number one is optical microscope. Optical microscopes use visible light to magnify specimens' image. Cells, tissues, and microorganisms, as well as small structures and particles, can be visualized using them. The second one is electron microscope. These microscopes do not use light, but still they achieve a higher magnification than optical microscope, and they are frequently used to examine the ultrasound of cells and tissues. Scanning probe microscopes. in order to image surfaces and material with extremely high resolution scanning probe microscopes use a physical probe to scan the specimen's surface. What is the method of microscopy? Prior to any procedure, as I mentioned, we will collect the samples. The specimen is collected. After the collection of the specimen, stain the specimen using the appropriate procedure. For example, gram stain or acid-fast stain. If bacteria are seen in gram stain, then their shape, for example, cocci, rod, size, arrangement, for example, chains or cluster, and whether they are gram positive or negative or acid-fast are observed. And it is also important to know the number, either one or more than one type of bacteria or more than one bacteria involved. Microscopy has advantages of taxonomic classification because when we go for staining in gram stain, we will get to know either it's gram positive or is gram negative. And in acid fast, we'll get to know which bacteria is it. I've got a detailed video on bacterial taxonomic classification. Its link is in the description or maybe in the top right corner. And also empiric therapy is done. What is empiric therapy? It refers to antibiotics that are administered during the period prior to receipt of blood culture and antibiotic susceptibility test results. There is only one disadvantage that microscopy has, and it is that it is not sufficient to identify an organism. Culture, a process for identifying and describing the species of bacteria in laboratory-grown samples. There are some terms that I would like to mention prior to telling you about the method of how to do culture. Number one is medium. Its plural is media. It is any preparation that contains nutrients essential for bacterial growth. As in this picture, you can see this is one medium, this is another, and these three together are called media. And they've got essential nutrients that are required for bacterial growth. Culture medium, a medium that has been successfully inoculated with bacteria. Inoculation of media. It means introduction of infected material to the medium for cultivation of organism present in that material. Major types of culture media. One is selective and the other one is differential. The selective media containing compounds that only allow certain bacteria to grow. For example, antibiotics, salts, or dyes. On the other hand, differential media is differential because they contain other compounds that allow only one type of bacteria to be distinguished from another based on biochemical reaction. For example, detecting hemolysis on blood agar plates or pigment formation. But in some places, you'll also find the classification of culture media based on physical state and composition. According to physical state, culture media is classified into liquid and solid. Liquid media is fluid in nature and it is usually placed in test tubes. It is also called broths. For example, nutrient broth, tryptic soy broth, phenol red carbohydrate broth. And solid media is prepared by adding solidifying agents like gelatin and agar to liquid medium. For example, nutrient agar, blood agar, mechanical agar, and chocolate agar. And according to composition, culture media is classified into simple media, which contains only basic substances such as nitrogen, carbon, and minerals. For example, nutrient broth, nutrient agar, peptone water. And the second one is enriched media. Enriched media is called enriched because it has got some enriched material like blood, serum, or aesthetic fluid. Aesthetic fluid is actually the fluid present in abdomen during ascites, mainly related to liver conditions. And this fluid, maybe serum, aesthetic fluid, or blood, is added to the medium required for proper growth of some bacteria. For example, blood agar, chocolate agar. The third one is differential media. We've discussed that. We've discussed selective media. The example of differential media is mechanical medium, and the selective media is Lowenstein-Jansson's medium. The final one is media for biochemical reactions. It is used to detect different biochemical reactions produced by different organisms. For example, thiamine citrate medium. Now we are going to have a look at commonly used bacteriologic agars. There are 3 columns in this table. First one is showing the name of the agar. Second one, it will be telling which bacteria is isolated on that agar. And the third one is telling the function or properties of the agar. I'll explain just 2 or 3 and we'll leave the rest to you guys. It's not difficult, don't worry. First agar is blood. Various bacteria are isolated in that agar and it is used to detect hemolysis. The second one is, it is an Italian word. It is Bodet Gango. I don't know how it is pronounced because I don't know Italian language. And I found that on internet that it is pronounced Django at some places and Gango at some places. And it is used for the isolation of Bordetella pertussis bacteria, and it is used to detect increased concentration of blood, which allows the growth. The third one is charcoal yeast extract. The bacteria isolated on that agar is Legionella pneumophila, and it has got an amazing property that increased concentration of iron and cysteine allows growth. Let me have a look at another one. Okay, let's look at Lowenstein-Janssen. The Mycobacterium tuberculosis is isolated on that agar. It selects against gram-positive bacteria in respiratory tract flora and contains lipids required for growth. Let's start talking about the cultures. The first one is blood cultures. It is performed in the following cases: sepsis, endocarditis, osteomyelitis, meningitis, and pneumonia. The bacteria most frequently isolated from blood cultures are 2 gram-positive cocci. The first one is Staphylococcus aureus, the second one is Streptococcus pneumoniae. And 3 gram-negative rods. The first one is Escherichia coli, the second one is Klebsiella pneumoniae, and the third one is Pseudomonas aeruginosa. Certain pathogenic fungi, including yeast, Candida species, and Cryptococcus neoformans and molds, can also be isolated from blood cultures. But we are not talking about fungus in this video because this is dedicated to bacterial lab diagnosis. The method of blood cultures. For blood cultures, the site for venipuncture must be cleansed by an antiseptic to prevent contamination by members of the flora of skin, usually Staphylococcus epidermidis, and decrease the risk of infection-related complications. The blood obtained is added to a rich growth medium in a bottle that contains an indicator for carbon dioxide production. Standard practice is to inoculate 10 ml of blood in each of 2 bottles per culture set, with 1 bottle incubated anaerobically and 1 bottle aerobically. Production of carbon dioxide within the bottle indicates that organism metabolism and growth have occurred. Once growth occurs, gram stain, subculture, and antibiotic sensitivity tests are performed. In some hospitals, molecular methods are also used to identify the organism. Next ones are the throat culture. Throat cultures are performed in diphtheria, pharyngitis, gonococcal pharyngitis, and oral thrush caused due to Candida. And they are used to detect the presence of group A beta hemolytic streptococci, the Streptococcus pyogenes. Gram stain is typically not done on throat swab because it is impossible to distinguish between the appearance of normal flora streptococci and Streptococcus pyogenes. Method. When the specimen is obtained, the swab should not touch only the posterior pharynx but also both tonsils and tonsillar foci as well. Material on the swab is inoculated onto a blood agar plate and streaked to obtain single colonies. If colonies of beta hemolytic streptococci are found after 24 hours of incubation at 35 degrees Celsius, a bacitracin disc is used to determine whether the organism is likely to be a group A streptococcus. If growth is inhibited around the disc, it is group A streptococcus. If not, it is a non-group A beta hemolytic streptococcus. Sputum cultures. These are performed in the cases of pneumonia, pulmonary tuberculosis, and lung abscess. And they are commonly used to detect Streptococcus pneumoniae, Staphylococcus aureus, Klebsiella pneumoniae, Pseudomonas aeruginosa, Mycoplasma, and Legionella pneumonia. Method. Prior to doing anything else, we are going to collect what? Samples. The sample of sputum is collected. It is the sputum, not the saliva or nasopharyngeal secretions from the upper airway. Then it is streaked on blood agar plate and the presence of colonies on the plate is revealed. Culture of sputum on blood agar can reveal the presence of colonies with identification established using various serologic or biochemical tests or by MALDI-TOF. It is abbreviated as matrix-assisted laser desorption or ionization time of flight. Cultures of mycoplasma are infrequently done. Diagnosis is usually confirmed by a rise in antibody titer. If Legionella pneumonia is suspected, the organism can be cultured on charcoal yeast agar, which contains the high concentrations of iron and sulfur which are required for growth. If tuberculosis is suspected, an acid-fast stain is done immediately and the sputum cultured on special media, which are incubated for at least 6 weeks. In diagnosing the aspiration pneumonia and lung abscesses, culturing for anaerobic bacteria is important. CSF cultures or cerebrospinal fluid cultures, these are performed in the following cases. If it is bacteria responsible for causing the disease, and diseases are meningitis, meningoencephalitis, transfer myelitis, CSF specimens from tissue-centric cases including encephalitis, brain abscess, and subdural empyema may show negative cultures. And for one fungus, and that is Cryptococcus neoformans. They are commonly used to detect these encapsulated organisms: Neisseria meningitidis, Streptococcus pneumoniae, and Haemophilus influenza. Method. Prior to doing anything else, sample is collected, then it is centrifuged, then what happens, it is taken through a staining procedure, either gram stain or acid-fast stain. Then the gram stain smear of the sediment of the centrifuged sample guides the immediate empirical therapy. The fluid should thus also be cultured on special media, and the culture is held for a minimum of 6 weeks. And molecular methods are also used to identify these organisms, and we are going to discuss what are these methods at the end of this video. And for the fungus Cryptococcus neoformans, it is a cause of meningitis, particularly in human immunodeficiency virus-infected patients. The India ink test was performed in the past, but in present, most labs use the latex agglutination test. That is for the Cryptococcus neoformans. It's cryptococcal antigen, and that is done on CSF as a more specific and sensitive test. Stool cultures. These are performed in severe and persistent diarrhea, nosocomial diarrhea, and enterocolitis, and also the bloody diarrhea. It is commonly used to detect Shigella, Salmonella, Campylobacter E. coli, especially its 0157 strains, and Clostridium difficile. The method is that feces are collected, specimen is processed within 2 hours, and plates used for different media are meconic agar, eosine methylene blue agar, Camphor CVA, and Cicro medium, meconic sorbitol medium, and also molecular methods are used for these bacteria that are present in feces responsible for causing diseases. Urine cultures. Urine cultures are performed in following cases like pyelonephritis and cystitis. And these are commonly used to detect Escherichia coli, which is the common cause of UTI, then Enterobacter, Proteus, and Enterococcus faecalis. Method is to obtain a specimen in a container or by using suprapubic aspiration and catheterization. Let me tell you something really cool here. Urine in the bladder of a healthy person is sterile, but it acquires organism of the normal flora as it passes through the distal portion of the urethra. To avoid these organisms, a midstream specimen avoided after washing the external orifice is used for urine culture. In special situations, suprapubic aspiration and catheterization may be required to obtain a specimen. How we are gonna form the urine cultures? We'll go for a calibrated loop that holds 0.001 ml of urine and can be used to streak the culture. The second one is serial tenfold dilutions that can be made and sampled from dilution streaked. And the third one is a screening procedure. It involves an agar-covered paddle that is dipped into the urine. After the paddle is incubated, the density of the colonies is compared with standard charts to obtain an estimates of concentrations of the bacteria in the urine. Something important about urine cultures. Culture to be done within first hour of collection. Urine to be stored in refrigerator for not more than 18 hours at 4 degrees Celsius. It is commonly accepted that a bacterial count of at least 100,000 per ml must be found to conclude that significant bacteria is present in the urine and in an asymptomatic patient. There is evidence that a bacterial count as low as 1,000 per ml is significant in symptomatic patients. Genital tract cultures. These are performed in the cases of abnormal discharge, STDs, sexually transmitted diseases, non-gonococcal urethritis, and cervicitis. And they are commonly used to detect Neisseria gonorrhoeae and Chlamydia trachomatis. The method is to obtain a specimen, and it is obtained by swabbing. By swabbing the urethral canal for men and cervix for women, and anal canal for both men and women. And then it is streaked on the plate and is left for the growth, and when the bacteria grows, and here we go for our diagnosis. Plates being used in genital tract cultures. For Neisseria gonorrhoeae, the Thay-Martin chocolate agar plate is used, and for Chlamydia trachomatis, the cultures of human cells or the cultures of yolk sacs of embryonated eggs are used. Because the Treponema pallidum, the agent of syphilis, cannot be cultured, diagnosis is made primarily by serology and sometimes by microscopy. Wound and abscess cultures. These are performed in the following cases: in the brain abscesses, lung abscesses, and abdominal abscesses. And in case of wound infections, traumatic open wound infections, surgical wound infections, and human bites. And they are commonly used to detect the following bacteria: Bacteroides fragilis, gram-positive cocci, S. aureus, S. pyogenes, Clostridium perfringens, Staphylococci, Streptococci, Propionibacterium acnes, Viridin streptococci, Streptococcus aeruginosus, Provotella, and Fusobacterium. The method is to collect a specimen in the collection tubes and then transport it to the culture and then streak that over the culture plate. When culture is negative, immunologic or molecular methods are used. Immunologic methods are which one? The serologic methods. How to diagnose a bacterial infection when the culture is negative? Detect the antibody in the patient's serum. Detection of immunoglobulin, IgM antibody, indicates a current infection. A single IgG antibody titer is difficult to interpret because it is unclear whether it represents a current infection or the previous infection. In certain diseases, a single titer of sufficient magnitude can be used as presumptive evidence of a current infection. Another thing that we can do when the culture is negative is to detect antigen in patient specimen. Use known antibody to detect presence of antigens of the organisms. For example, fluorescent antibody to detect antigens in tissue. Latex agglutination to detect capsular polysaccharide antigens in spinal fluid. The third thing that we can do when culture is negative is to detect nucleic acids, definitely in the patient's serum, and then use polymerase chain reaction, the PCR, and DNA probes to detect the DNA or RNA of the organism. And now we are done with the cultures. Let's talk about serologic methods or serology. It is the process to detect antibodies in a patient's blood that are specific to bacteria causing the infection. There are two basic approaches to that. Number one is using known antibody to identify the microorganism, and the number two is using known antigen to detect antibodies in the patient's serum. Methods. There are two methods to do that. Number one is identification of an organism with a known antiserum. The following four tests are involved in this one. The first one is side agglutination test. It is the antisera that can be used to identify Salmonella and Shigella by using agglutination, meaning clumping of unknown organism. The second one is latex agglutination test. The latex beads coated with specific antibody are agglutinated in the presence of homologous bacteria or antigen. This test is used to determine the presence of capsular antigen of the yeast Cryptococcus neoformans. The third one is ELISA, E-L-I-Z-A, and it stands for enzyme-linked immunosorbent assay. In this test, a specific antibody to which an easily assayed enzyme has been linked is used to detect the presence of homologous antigen. This test is useful in detecting a wide variety of bacterial, viral, and fungal infections. The fourth one is fluorescent antibody test. A variety of bacteria can be identified by exposure to known antibody labeled with fluorescent dye, which is detected visually in ultraviolet microscope. The second method in serologic methods is identification of serum antibodies with known antigens. It includes side or tube agglutination test. In this test, serial two-fold dilutions of a sample of patient serum are mixed with standard bacterial antigen suspension. The highest dilution of the serum capable of agglutination is the titer of the antibody. As with most tests of the patient's antibody, at least a four-fold rise in the titer is actually the concentration of antibodies in the blood. At least a four-fold rise in the titer between the early and the late samples must be demonstrated for a diagnosis to be made. This test is used primarily to aid in the diagnosis of typhoid fever, brucellosis, plague, leptospirosis, and rickettsial diseases. The second method is serologic test for syphilis, as I mentioned earlier. It is a detection of antibody in patient serum and is frequently used to diagnose syphilis because Treponema pallidum does not grow on laboratory media. There are two kinds of tests. Number one is the non-treponemal tests, and the second one is the treponemal test. I'm not going to go in detail because this will make this video too complex, guys. Okay, now have a look at the cold agglutination test. Patients with Mycoplasma pneumoniae infections develop autoimmune antibodies that agglutinate human red blood cells in the cold at 4 degrees Celsius but not at 37 degrees Celsius. These antibodies occur in certain diseases other than Mycoplasma infection, thus false positive results can occur. Finally, we are going to talk about molecular methods. Molecular tests can detect the presence of bacterial DNA, RNA, or protein in patient specimens. These tests are both sensitive and specific, and they've become the diagnostic gold standard for many infections. There are two types of molecular methods. First one is genomics test, that includes nucleic acid amplification test, and the second one is proteomic test, that includes the matrix-assisted laser desorption or ionization time of flight. The specificity of these tests resides in the ability of the DNA or RNA probe to bind to DNA or RNA present only in the bacteria to be identified. I know it was too much knowledge in today's video, so I'm going to review that by summarizing it here. How to diagnose a bacterial infection or the diagnosis of bacterial infection. Prior to doing anything else, we are going to do what? We will obtain a specimen from the infected site. Then we will go for microscopy. We'll stain the specimen either with gram stain or acid bath stain. And if bacteria are seen, then we will identify the type, either it's gram positive, negative, or acid fast, then its number, shape, size, and arrangement. And then we'll go for the culture. Bacterial samples are grown in laboratory to identify and characterize the bacterial species. There are two types of the culture, that is the selective and differential. For example, urine culture, stool culture, and blood culture. Serology. It detects antibodies in a patient's blood that are specific to bacteria causing the infection. Molecular methods. These detect the presence of bacterial DNA, RNA, or protein in patient specimen. And that's it for today's video. I hope you guys enjoyed it. You've learned something. If you've got any suggestions, feel free to leave them below in the comment section. And also, if you want to connect with me on my social media, I've got my Instagram, I've got my Twitter, and I do upload blogs. I'll catch you soon. Till then, Assalamualaikum.